Welcome back, fellow mitochondriacs, for another episode of Cancer as a Mitochondrial Metabolic Disease. As you may or may not know, last week's live stream, when trying to talk about the Thioredoxin system, did not work out as planned. Unfortunately, it said there was a poor connection despite me having Ethernet connected to my computer. Anyways, we're going to attempt this a second time. And I think that considering everything in this particular slide set flows together, it would make sense to just kind of start from the top again and start to look at these other antioxidant systems that you may or may not be aware of. So we're basically in a series of oxidative therapies. And in order to understand how oxidative therapies are either effective or not effective, or how to maximize their effectiveness, we have to understand what we're up against. And I think that up until now, the major antioxidant system that I have discussed on this channel happens to be glutathione. And glutathione is a major system. And we left off on this slide here in the prior video, recorded video, looking at how both glucose and glutamine effectively contribute to the glutathione pool and the glutathione recycling ability of these cancer cells to either put these cancer cells either in a ferroptosis resistant or a ferroptosis sensitive state. But although glutathione is a major antioxidant contributor, it's not the only antioxidant contributor. And we had kind of touched in the last video or the beginning of the last live stream that got cut off 10 minutes into the 50-minute that I presented. We started talking about other antioxidant systems. In particular, we started to highlight the thioredoxin system. And we then went to this paper here, which is titled Exploring the Thioredoxin System as a Therapeutic Target in Cancer, Mechanisms, Implications. And it says that one key component of this defense is the thioredoxin TRX system, which includes TRX, thioredoxin reductase, TRXR, and NADPH. And I quickly highlighted how NADPH is provided to us, at least in part, a major part, by the PPP, the pentose phosphate pathway, which requires glucose 6-phosphate or glucose as the major first substrate for that system. Although it's really never mentioned in these papers about how glucose contributes to this system, just know that when we see NADPH here, we know that glucose is at least partially responsible for the recycling of this system through NADPH. And so therefore, glucose is also an important part. So what I'm trying to say is that being on a ketogenic diet or inhibiting glycolysis on the kind of the higher levels of glycolysis before it hits glucose 6-phosphate, basically GLUT transporters as well as the hexokinase enzyme will have an effect on the system as well. The TRX system reverses oxidation of macromolecules and indirectly neutralizes ROS via the pyridoxine PRX. This dual function protects cells from damage accumulation and supports physiologic cell signaling. However, the TRX system also shields tumors from oxidative damage, aiding in their survival. Due to elevated ROS from their metabolism, tumors often rely on the TRX system. In addition, the TRX system regulates critical pathways such as the proliferation and neoangiogenesis, or the creation of new blood vessels, which tumors exploit to enhance growth and optimize nutrient and oxygen supply. And what we see here is that, the TRX system, you would imagine only has to do with basically the antioxidant defense or maybe the protection of the DNA through, again, antioxidant defense. But it has a lot of other things that it is responsible for through allowing these cancer cells to tolerate higher levels of reactive oxygen species. And some of those higher levels of reactive oxygen species is what leads to a lot of these issues here through the hypoxia-inducible factors and so on and so forth. We left off on this particular slide during the failed attempt of the live stream, and I was reading off this particular piece of the paper, and I was talking about how the primary cellular ROS or reactive oxygen species within cells include superoxide, O2, negative hydroxyl radical, OH, and hydrogen peroxide. In general, ROS production begins with electron transfer to oxygen, forming the superoxide radical, which is highly reactive, with a short half-life of about 1 microsecond, and has limited diffusion through membranes due to its charge. In aqueous environments, O2 rapidly converts to hydrogen peroxide, and although hydrogen peroxide is not a true radical, it is considered as a reactive oxygen species compared to other ROS that has a longer half-life, about 1 millisecond, and can diffuse through cell membranes and is rather specific, oxidizing primarily free thiols. And I'm pretty sure this is where the video got cut off. Yes, thiols are what make up glutathione or the thioreductase systems. These are sulfur, basically amino acids or sulfur atoms that are contributing to the redox homeostasis of these molecules. So basically, what it's saying here is that this hydrogen peroxide primarily will kind of be interacting with these sulfur molecules. And that brought up a couple of questions, even in our little truncated video that we did last week. Should you avoid sulfur containing compounds or sulfur containing amino acids? And I'm not going to completely avoid the discussion because I'm going to have an entire video or maybe even a couple of videos about basically what substances or compounds or supplements would probably be best to be avoided when you have cancer and you're trying to utilize either conventional therapies such as chemotherapy or radiation therapy or ketogenic metabolic therapies and ways that we could potentially kind of blunt progress. And one of the ways we can do that is by taking exogenous antioxidants. And in particular, maybe even the certain amino acids that make up antioxidants, such as glutathione, which is relying on cysteine, a sulfur-containing amino acid, and glycine and glutamine and or glutamate, which is being converted from glutamine to make glutathione. And then the other question is, what about sulfur-containing compounds such as MSM? And I was able to find, and I'll talk about this in the near future when I have a dedicated video about this, because this is a question I get over and over and over again, rightfully so. And what I was able to find was that MSM in particular can likely worsen outcomes when you have cancer based off the fact that it's able to donate those sulfur molecules to these systems. I would also say that probably methionine, which is a sulfur-containing amino acid, is probably also a no-no, at least supplemental methionine. I mean, of course, there's probably not a lot we can do from like restricting per se amino acids in food, although, a ketogenic diet is not a high protein diet. But that being said, I think there are kind of entire metabolic theories of cancer that have to do with methionine. And although I don't believe methionine is used to make energy, I do believe that the methionine potentially contributes to redox homeostasis. And that's maybe why giving extra, methionine would be bad. And potentially by, inhibiting it or, using methionase, so on and so forth, could potentially be in our favor or our benefit because it's effectively cutting off the antioxidant capacity of the cell. I'll digress on that until we get to those videos about what not to take when you have a diagnosis of cancer. I do want to, take a second here and just reiterate that these systems are our endogenous systems that we use to protect ourselves from reactive oxygen species and oxidative stress. So I'm not trying to say that the glutathione system or the thioredoxin system or other antioxidant systems are bad. They're not either good. They are a tool our body uses. But the problem is that these tools are effectively hijacked by cancer cells, which use that system against us. So therefore, we're going to understand they're using that system against us and try to use that system against them. Hydrogen peroxide can be converted into water. This occurs via enzymes such as catalase, which we'll talk about in a future video, which transforms hydrogen peroxide to O2 and water. Another key factor in hydrogen peroxide removal is the tripeptide glutathione, which acts as an effective oxidative scavenger in its reduced form, GSH. In addition to GSH, thioredoxin, TRX, acts as an essential disulfide reductase, reversing thiol oxidation. TRX is an important component of the cellular redox system, playing a crucial role in regulating various redox signaling pathways. It is also increasingly recognized as an important modulator of tumor development and tumor growth. The thioredoxin system, comprising of NADPH, remember glucose, PPP, etc., TRX, and thioredoxin reductase, is highly conserved in all eukaryotic cells. It plays a crucial role in regulating various cellular functions, including the activity of transcription factors, DNA synthesis as a hydrogen donor and ribonucleotide reductase, and cell growth through the activation of kinase cascades. The functions of the thioredoxin system. The TRX system reverses protein oxidations and can scavenge ROS, thus maintaining cellular redox equilibrium and modulating oxidative signals. Oxidative signals are integral to many signal transduction cascades, with hydrogen peroxide playing a key role due to its relatively long half-life and its ability to diffuse through biologic membranes, and its primary attack on free thiols against sulfur molecules, leading to disulfide bond formation. The reversibility of such oxidations is crucial for signaling, thus TRX being pivotal for reducing disulfide bonds, therefore enabling the switching on and off of these signals. And we have a schematic here of basically the thioredoxin system. So we have an oxidized protein, and that protein is going to get reduced by this thioredoxin system. And then thioredoxin has to be basically recycled by this thioredoxin reductase. And that requires NADPH. Again, glucose is contributing through the PPP through this system. So although this thioreductase is a target and thioredoxin is a target, being on a ketogenic diet is affecting this process as well. And then thioredoxin can also interact with this pyroredoxin as well. So this is just kind of showing how this thioreduction system can be interacted by inflammation and how that and ROS can contribute to the upregulation of this thioredoxin system. And this is just another schematic showing how both reactive oxygen species, oxidative stress, and hypoxia can also lead to this thioredoxin system being activated, working through hypoxia-inducible factor and NRF2, which is the antioxidant response element, ARE. I'm guessing hypoxia response element as well, and how that leads to both the continuation of the Warburg metabolism, the metabolism of glucose and glutamine, but as well as the progression of cancer through angiogenesis, migration, invasion into other tissues, and the continuance of the cancer system. So the thioredoxin system in cancer, the TRX system, is undoubtedly a double-edged sword. On one hand, it protects against oxidative damage, preventing the oxidation of macromolecules such as proteins, lipids, and nucleic acids. This protects cells from the accumulation of mutations, thereby defending the organism from the development of malignancy. So therefore, it has a protective effect at helping protect us from getting cancer. But once we have cancer, it's effectively used against us. An existing malignoma or an existing cancer benefits from these protective mechanisms. Tumors often exhibit elevated ROS due to their metabolism. This TRX prevents this increased ROS production from damaging and killing tumor cells. This increased proliferation due to the activation of transcription factors such as NF-kappa B or AP1 leads to faster tumor growth. Additionally, hypoxia-inducible factor 1 alpha significantly promotes angiogenesis and metastases formation. Activated HIF1 alpha induces various target genes such as vascular endothelial growth factor, VEGF, and matrix metalloproteinases, MMPs. Genes VEGF promotes angiogenesis, while MMPs facilitates invasions and metastasis. There's a correlation between the overexpression of components of the TRX system and multi-drug resistance, MDR. In pancreatic cancer cells, higher TRX expression protects against cisplatin-induced apoptosis. In KRAS wild-type colorectal cancer cells, increased TRX-R2 expression is involved in the development of multi-drug resistance, MDR. It has been shown that the TRX expression is increased to protect various cancers, leading to increased tumor aggressiveness and poorer overall survival, including in colorectal carcinoma, CRC, lung cancer, pancreatic cancer, gastric cancer, hepatic cancer, breast cancer, and tumors of the hematopoietic system, basically lymphomas, leukemias, etc. I would dare say that this is in play for all cancers, given there's such a diverse ones that are listed here. A correlation between the thioredoxin system and resistance to paclitaxel has also been observed. In ovarian cancer cells, it has been shown that TRX binds to the transcription factor fork head box protein 01, FOXO01, and induces its translocation to the nucleus and therefore promotes the expression of FOXO1 dependent genes that protects the tumor from cell death. TRX can also protect breast cancer cells from tamoxifen-induced apoptosis by directly scavenging hydrogen peroxide via PRX or inducing estrogen-dependent and estrogen-independent redox-sensitive survival pathways. Increased expression of the TRX system proteins also is associated with resistance to doxorubicin, docetaxel, and tamoxifen. Therefore, the overexpression of TRX system components not only results in more aggressive, rapidly growing, and highly metastatic tumors, but also significantly limits the treatment options and reduces patient survival. Thus, the individual components of the TRX system represents viable targets for cancer therapy. I will just add my own little commentary here. It says that it significantly limits treatment options and reduces patient survival. It significantly limits conventional treatment options, right? Because our treatment options of ketogenic metabolic therapy will address this system, at least in part by restricting glucose to these tumors, aka NADPH, aka able to recycle this TRX system. And some of the other metabolic therapy components that we commonly utilize will also be at play here, and we'll see that very shortly. Manipulation of the thioredoxin system, potential therapeutic approaches. The TRX system is essential for maintaining cellular redox balance and, together with PRX, acts as a crucial ROS scavenger. On one hand, it helps control oxidative signaling pathways, prevent oxidative damage, and reduce mutation accumulation, thus ensuring normal cell function and preventing cancer. On the other hand, the TRX system protect established tumors from cell death and even promote their proliferation. Cancer cells often exhibit higher ROS levels, and the TRX system protects these cells from oxidation-induced cell death. Additionally, since the TRX system regulates proliferation promoting transcription factors, it can further enhance the proliferation of malignancies. Inhibition of thioredoxin. Inhibiting TRX presents a promising target. Particularly interesting are the cysteines and the active site cis32, cis35, as well as cysteine cis 62, 69, 73, which, although not located in the TRX activity, those are less important for us to know the locations of those cysteine residues, et cetera. So what we have is we have a couple of compounds that are able to interact with this TRX system in a variety of ways. And if you remember, when I actually read the abstract on the live stream that did not go very far, I read about PX12 and DMF, which also stands for dimethyl fumarate, which are the two that are listed on this particular graphic. So it says here that schematic representation of the mechanism of action of PX12 and dimethyl fumarate. A, the top picture. PX12 inhibits thioredoxin 1. This leads to the accumulation of reactive oxygen species, which oxidizes macromolecules such as lipids, proteins, and nucleic acids. These oxidative damages, particularly the accumulation of DNA damage, induce the intrinsic apoptosis cascade. The pro-apoptotic BCL2 family members, BAX, BCL2 associated X protein, and BAK, BCL2 homologous antagonist killer, oligomerize and form a pore in the outer mitochondrial membrane, leading to the collapse of the mitochondrial membrane potential and release of cytochrome C. This release of cytochrome C induces the formation of the death platform called the apoptosome and the induction of cell death. In the second picture, B, DMF, also inhibits TRX1. As a result, the redox-sensitive transcription factor NF-kappa B can no longer be reduced in the nucleus. NF-kappa B can no longer effectively bind to its binding site in the promoter regions of target genes. This leads to the decreased expression of anti-apoptotic proteins CFLIP and the inhibitor of apoptosis IAP proteins IAP1, 2, and XIAP. The absence of IAPs allow for the formation of the death platform called the riptoptosome. This can induce the both apoptosis and necroptosis through the receptor interacting serine threonine protein kinases RIP1 and RIP3 if the apoptosis pathway is blocked. And that's what we're seeing here. We're seeing DMF added to the cell. The DMF will then interact with the TRX1, again, increasing reactive oxygen species, just like we saw in this past one, but also interacting with TRX and its ability to interact with the NF-kappa B. And then that allows for these anti-apoptotic proteins not to be created, which allows for apoptosis and necroptosis. Now, PX12 and DMF may or may not be available to us, but what is available to us that interact with the system? Well, what about sodium butyrate? Sodium butyrate is a short-chain fatty acid mainly produced by colonic bacteria. It has been shown to reduce the activity of TRX1 in colonic carcinoma cells or colon carcinoma cells, while the activity of TRX1 in healthy mucosal did not change after sodium butyrate treatment. I think this is probably one of the most amazing things, is that somehow these compounds know that there's a disease cell and they will have an opposite effect, or at least a negligible effect, on the normal cells, but on the cancerous or disease cells, they have their effect that leads us to have the ability to overcome these cells. I think it's unbelievable. Sodium butyrate inhibits HDAC activity, acting as an HDAC inhibitor, histone deacetylase, leading to the histone hyperacetylation, similar to that of SAHA. Sodium butyrate induces the expression of the natural TRX inhibitor, TXNIP, which restricts TRX activity. These results were also confirmed in vivo studies where treatment with sodium butyrate led to the reduction in tumor growth. The next compound that we probably have access to is the diallyl trisulfide. Diallyl trisulfide, also known as alitridin, is an organosulfur compound derived from garlic. It can be formed through the hydrolysis of allicin. Through Michael addition, diallyl sulfide directly conjugates with this cysteine 32 and cysteine 35 residues in the active site of the TRX1, therefore inhibiting it. The predominant cause of high mortality in triple-negative breast cancer, TNBC, is metastases. It is suggested that TRX1 plays an important role in breast cancer metastases. Analyses of nude mice model of spontaneous and induced breast cancer metastases show that diallyl trisulfide leads to the inhibition of TRX1 and suppresses metastases. In addition, it has been demonstrated that diallyl trisulfide sensitizes glioblastoma cells to radiotherapy. This is likely directly related to the inactivation of the TRX1 and weakening of the oxidative defense by diallyl trisulfide. I personally don't like when they reduce one particular awesome compound like garlic or sodium butyrate to one particular mechanism of action that leads to their success and inhibiting certain types of cancer. But I do think that in this case, it is important to note that there's at least one important way that this particularly works, and that is the inactivation of these antioxidant systems, particularly the TRX system. And I think it's pretty interesting that it says that it will then sensitize glioblastoma cells to radiotherapy. Why would that be? Because radiotherapy is an oxidative therapy. And so when you are using an oxidative therapy, if there is plenty of reactive oxygen species scavenging proteins or antioxidant systems available to the cancer cell, then those systems will be able to basically overcome any oxidative stress or the majority of the oxidative stress that is being induced by whatever particular oxidative therapy you are utilizing, in this case, radiation therapy. So it makes total sense that it would sensitize whatever cancer to radiation therapy because that's how radiation therapy functionally works. Again, going back to a slide that I like to go over and over and over again. So basically, when we have, this is particularly talking about glutathione, but my point by showing this is that basically we have cancer cells that are in these resistant states, high antioxidant systems in place that allows them to be in a basically cell death resistant state, in this case, ferroptosis. And so when we nix the TRX system and we shoot it with some kind of an oxidative therapy, we are in a ferroptosis sensitive state. So it allows that particular therapy to be more effective. In this case, we're talking specifically about radiation therapy, but that could be likely extrapolated to other oxidative therapies that we will talk about in this series. Another compound that is at play here is a compound called renofolin. And this is a compound that I have a very difficult time pronouncing. So in the literature, it's also known as AF. So from now on, I'm just going to use the word AF so I don't continue to butcher this particular name that I have trouble pronouncing. So AF inhibits thioredoxin reductase 1, TRXR1, leading to reactive oxygen species ROS accumulation. Elevated ROS can cause the accumulation of misfolded proteins in the endoplasmic reticulum, triggering stress and the induction of the unfolded protein response, UPR, which can induce cell death. ROS also causes DNA damage, activating the intrinsic apoptosis pathway. This results in the mitochondrial outer membrane permeabilization, MOMP, and the release of cytochrome C from the mitochondrial intermembrane space through the pore formed by BCL2 associated X protein, BAX, and the BCL2 homologous antagonist killer, BAK, subsequently leading to the formation of the apoptosome and induction of apoptosis. AF further exacerbates this by inhibiting mitochondrial thioredoxin reductase 2, TRXR2, causing mitochondrial damage, including to the electron transport chain, which increases ROS production and oxidative stress. So what we have here is AF acting on multiple TRX reductase systems. So we have TRX1, which is directly relating to ROS production. That leads to stress and DNA damage. Basically, in another way of activating the apoptosis system. And then TRXR2 is in the mitochondria, which is allowing for mitochondrial ROS to be at least curtailed and not completely destroying the mitochondria. That leads to an increase in mitochondrial damage, the ejection of cytochrome C, which then allows the apoptosome to be formed through the caspase system and the apoptosis through that pathway as well. So in the past mechanism, we're looking at the TRX1 protein, and this we're looking at the TRXR protein. And let's talk about some things that we have potentially access to for this. So curcumin is one of my favorite nutraceuticals, probably of all time, because it has so many effects on these cancer systems. Curcumin is extracted from Curcuma longa, a plant species belonging to the ginger family. Anti-inflammatory and anti-cancer properties are attributed to curcumin. It has been reported that curcumin inhibits, among others, NF-kappa B and TRXR. Curcumin hinders the activity of TRXR by binding to this active site and disrupting the electron transfer from NADPH to TRXR. Several studies show that curcumin leads to the accumulation of ROS in tumor cells, inhibiting metastases formation, and induces cell death and/or sensitizing cells to radiation therapy. This is a great place to just reiterate this. There are plenty of ways within normal cells where curcumin will activate the NRF system and bolster, I'll repeat, bolster the antioxidant response. Most people believe curcumin to be an antioxidant, but curcumin really just upregulates the NRF2 pathway, which is our endogenous antioxidant response element. However, curcumin, as well as many other things like quercetin or EGCG or etc., will have an opposite effect within cancer. And it shows here that curcumin leads to the accumulation of ROS in tumor cells, which ultimately leads to apoptosis, induced cell death, and sensitization of cancer cells to radiation therapy because it's inhibiting the cancer cell's ability to mount antioxidant response, at least through the TRXR system in this particular publication, as we've seen in other publications and other ways of looking at curcumin, it has diverse effects across glucose and glutamine, which affect the glutathione system as well, the NADPH created through the PPP, etc. It is diverse in its actions. In particular, we're talking about the TRX system in this particular case, but all of those things will lead to the sensitization of cancer cells to radiation therapy and other oxidative therapies as well. Curcumin exhibits limitations for potential clinical applications due to its poor water solubility. Therefore, analogs have been developed, such as WZ26, which inhibits TRXR and restricts the proliferation and survival of tumor cells. Currently, additional analogs are being developed and tested. Several clinical studies have already been conducted on curcumin. In a phase 1 trial with theracumin, a curcumin derivative that demonstrates higher bioavailability than curcumin, it was shown that a combination of irinotecan is safe and well tolerated in patients with advanced solid tumors. Another clinical study on patients with colorectal carcinoma and unresectable metastases demonstrated that a combination treatment with bevacizumab FOLFIRI with a ginsenoside modified nanostructured lipid carrier containing curcumin resulted in a 1-year survival rate of 87.4%. In a phase 2 clinical study, patients were treated with either FOLFOX or a combination of FOLFOX and curcumin. The combination therapy showed no significant side effects and was deemed safe. Although a slightly improved overall survival rate was observed, there were no significant improvements in quality of life or reductions in neurotoxicity compared to the FOLFOX treatment alone. In additional studies that have been completed but have yet released results, curcumin has been investigated for the treatment of colorectal carcinoma, prostate cancer, breast cancer. Another study on the treatment of leukemia has completed the recruitment phase, while studies on cervical cancer and prostate cancer are still in the recruitment phase. Initial results are expected in 25 and 26, respectively. Although this thioredoxin system, this antioxidant system, is not very well known to many people, it is a powerful antioxidant system that is endogenous inside of our bodies that helps protect against reactive oxygen species. The problem is this system is hijacked, like many other systems, by cancer and becomes one of our biggest hindrances to using conventional or non-conventional therapies against cancer. When we have the know-how of how cancer cells protect themselves from reactive oxygen species, it allows us to then ask the right questions of how do we minimize that system so that we can employ systems to overcome that system. And so therefore, it allows us to ask the right questions of how do we inhibit that system, which is preventing our conventional and non-conventional therapies from working, so that we can then allow those conventional and non-conventional therapies to have maximal effect. It's important to note that there are several drugs and developmental compounds that are used in these examples, but I wanted to, as I did in these papers, show that there are natural alternatives to these drugs that you probably may or may not already be using, which gives us more kind of real-time actionable steps that we can take with things that we have access to. If you like videos like this, please like, share, and subscribe. And until next time.